Good morning, everyone. Thank you. I'm so grateful to be here, and thank you, Samir, and thank you, Luna, for the invitation and the arrangement. Yeah. And today, Valentin and I will share the presentation. So we'll talk about we use the R230 system for the C. elegans high-throughput proteomics sample preparation. So here, just a disclaimer and also a COI statement. And yeah, I think Samir already escaped this. So I want to introduce our lab very quickly. So our NCATS, ACC, mass spectrometry team. So we actually can see we cover a very wide range of applications. So our main focus on 4 areas. So let me think. Yeah, it's time to work. If I can use my mouse, yeah. So we mainly focus on 4 major areas. So the first is a high throughput component screen. So we focus on this one. Okay, thank you, Valentin. Yeah. Okay, that's great. So we do biochemical assay, we do cell-based assay, we analyze small molecules, we do peptides, we do proteins. And the major instrument we use is called Rapidify, a very high throughput SPE system. So basically it's labor-free and it's automated. So the cycle time is about 7 to 10 seconds per sample. And if you bypass this SPE cartridge, we can realize 2 to 3 seconds per sample. It means each day we can complete thousands of compound screen. So the next major area our lab focuses on is multi-omics, especially for proteomics. So we do drug target discovery, system biology, and structural proteomics, and also bioinformatics. And today we will highlight the high throughput proteomics and also the automated sample preparation. We also run an antibody and also some high-res support for our medicinal team. So I think Jennifer did a very excellent job introducing proteomics. So I think probably I can skip part of this slides, but I just want to highlight. So proteomics plays very vital roles in translation science field. For example, like drug targets, and also the selectivity, drug affinity, and also the toxicity. And in clinical phase, like biomarker, and also the treatment response. So this is a traditional proteomics workflow. So we have like bottom up, middle down, and also top down proteomics. So today we are focused on the bottom up proteomics. And if you have the hands-on experience, so you will know for the protein bottom-up proteomics sample preparation, we have like a protein extraction and also the digestion step. And maybe just a quick survey, how many of you have the hands-on experience for proteomics sample preparation? Yeah, okay. Normally, if you manually process your samples, how many samples you can process each day? 20, 30? Yeah, that's probably, yeah, I want to share the story of what I did when I was a postdoc. So at Johns Hopkins. So this is a work I identified a biomarker for malaria in human saliva. And then I have to digest hundreds of samples to quantify the target. And the outcome is very fruitful. So we have a paper published on Science Translational Medicine. We also have associated patent license to our company to develop the prototype of RDT, diagnostic test. But something behind, the story behind is I have to process over 400 samples and during Christmas, because in that time, the only triple quad available time is January. So I have to complete all the samples. Valentin is nervous. Yeah, you are fine. We want to push you working during Christmas. The other reason is because after I joined NCATS, so we developed a high throughput sample preparation, proteomics sample preparation platform. So this, the major work was done by my previous postbac student, Mr. Drew Burns, now it's actually Dr. Drew Burns at UIC now. So here I list several proteomic sample preparation bottlenecks. The major part I want to highlight is low throughput, as I mentioned. Probably manually you can process 20, 30 samples a day. So here we proposed integrated high throughput system, which you can culture your cells in 3D form, like a well and a plate. And then we integrate all these steps from this plate, the cell lysis, reduction, and alkylation, precipitation, and final digestion, get the clean peptide. and then we can subject to the LC-MS analysis. So for this instance, so you guess how long it take? Yeah, 4 hours. We can process like over 300 samples. It means by one day we can complete larger than 700 samples. So this is directly from the cell culture plate. So and also feasible for drug treatment protocols. And we also make some, modify this system to for clinical samples. For example, serum samples. In that case, so after the high bound protein depletion, so we modify the protocol. In that case, we can complete like over 300 samples just in 3 hours. And also we try to adapt this system to multicellular kind of samples. For example, worm samples, C. elegans samples, but we select there. And because the protein extraction, the thick cuticle of the C. elegans. So we got to know Covaris, we know the R230 systems, and more likely we have Valentin here, so very talented scientist, so she will continue the story with you guys. Okay. Oh, did you turn it on?
Sorry. Perfect, sorry about that. All right, thank you, Dinian. So as Dinian introduced our high-throughput automated proteomic platforms, so at this stage, as we mentioned, we're able to process adherent cells and serum samples. And so where I come in now is want to take us further and try to target multicellular organisms, specifically C. elegans. And so the question first becomes, well, why C. elegans? C. elegans are one of the most widely used model organisms due to a lot of the characteristics that I've listed here on the slide. However, they do have some characteristics that make their application in high-throughput screening a little bit more challenging. But that is something that our collaborators at NCATS have already tackled, and they were able to devise a quantitative high-throughput screen to study this complex model organism over the course of development and several life cycles. And so where we come in, and where specifically I come in, is we want to extend this platform and provide omics-level characterization of these C. elegans to basically be able to better decipher the pharmacological effect on phenotypes in these quantitative high-throughput screens. The challenge now that we face is that C. elegans, as Dinian mentioned, has a very thick outer cuticle. So it makes extraction of biomolecules such as proteins a very non-trivial task. And so a lot of these traditional lysis protocols that I've highlighted here, from manual grinding to solvent extraction, are not very efficient. The reproducibility is definitely lacking, and none of these are amenable to high-throughput workflows. And so that's where the use of the AFA technology really became important for us. What we were specifically excited about is that this extraction could be done with no contact between sample and probe. It was done in a temperature control environment, and we could scale this up all the way to 384 well format. So it was definitely high throughput. So as a first pass, what I will mention is that, and what Samir very nicely showed, is that this technology had yet to be used for C. elegans, specifically extraction of proteins. So we had no idea if it worked, and we just tried it out for the first time. And we're very excited that we were able to show the first successful extraction of proteins from C. elegans. So our initial attempt was using this R230 instrument, which is capable of processing 8 well strips. And so in these 8 well strips, we had 40 worms per sample, and we're able to identify about 1,500 proteins per sample. So this is very exciting, and what normally would have taken hundreds to thousands of worms, we were able to do with 40 worm samples. We then moved to using the R230 instrument, which I had mentioned is capable of doing 8 well strips, 96 well plate, and 384 well plate. So as a first direct comparison, we took our 8 well strips, put 1 worm per well, and we're able to achieve really, really exciting results where we can see across the life stages, we can see up to about 2,000 proteins in our adults, and even down to L1 worm, which is about 100 nanograms of protein, identify about 1,000, be it with some reproducibility issues in this first pass. We were very excited about this, so we had worked out the kind of initial beginning parts of our workflow. where now at that stage we're still doing all the manual sample preparation. So we wanted to see, okay, we have the lysis step automated. Can we now incorporate our automated sample prep platform and do the reduction, alkylation, and digestion on the Bravo instruments? And so we did that, and we combined the two workflows, and just as a first pass, use the protocol we had established for our cultured cells for worm samples. And what we saw actually here was a decrease in overall protein identification, as well as a decrease in reproducibility. So that told us we need to optimize our system on using the Bravo system. So we optimized some of the digestion step, the amount of worms we were using, as well as some of the liquid transfer steps. And with all these optimizations, we now really saw a big increase in overall protein identification and a really strong increase in reproducibility, especially in these early life stages. So I will note we did have to go up to 3 worm per sample in these experiments. All these were done in 8-well strips. So the next question we asked ourselves is, can we push the boundary of throughput since the R230 instrument is capable of processing 384-well plate? So we harvested our worms directly in a 384-well plate, lysed them in the R230 instrument such that we could take that plate and put it directly on our Bravo platform without having to do an additional manual transfer step. And with that, we were really excited because we saw an overall 1.5-fold increase in protein identification across all life stages. Seeing this increase in protein IDs, we saw, all right, can we go back to our single worm sample? And even with those, we saw a 1.5-fold increase in our later life stages. Unfortunately, as most of us will know, we always run into some instrument snafus and stuff. So we did have some sample loss issues due to some mass spec errors. So unfortunately, I do not have any data for the early life stages, but I'm confident that we would see an increase in protein identification in these early life stages as well. And so at this point, our pipeline is capable of processing these C. elegans samples. We have reduced the overall processing time from 2 days to 8 hours. We're able to process up to 384 single to multiple worm samples simultaneously. But what we now run into the issue of is that one proteomic run takes about 120 minutes of mass spec acquisition time. So if we run an entire 384-well plate, that would take us over 32 consecutive days of mass spec time, not counting any blank runs or weekly calibration or instrument issues. So we need something to speed that process up. And what we turn to using are TMTs. So essentially with a TMT, you can label, in our case, up to 16 differentially treated samples, digest down to the peptides, label the peptides with the differential tags such that these 16 samples can be pooled together into one injection on the mass spec. But we find ourselves now in a 1-step forward, 2-step back situation because this workflow is typically done in manual fashion in Eppendorf tubes, requiring about 25 to 50 micrograms of protein. And what I had mentioned is that a single worm sample will give you about 100 to 200 nanograms of proteins. So we needed a way to miniaturize and automate this workflow into a 3-day-4-well plate, where essentially you can plate 1 sample per well, 1 TMT tag per row, such that a 384-well plate could be reduced down to 24 pooled samples to be injected on the mass spec. So we did that optimization using commercially available HeLa protein digest, and we're now able to label down to 100 nanograms of protein using a 1:100 peptide to TMT ratio. Applying now this platform to our life stage analysis of our C. elegans, what would take us about 33 hours of mass spec acquisition times was able to take us only 3.5 hours of mass spec time, and we see an overall improved reproducibility as well. Diving a little bit deeper into this life stage analysis, we actually see our expected increase in protein abundance across the developmental life stages. We see clustering of the life stages, IPCA. And when we compare our label-free to our TMT data, we actually see a very strong Pearson correlation. And about 96% of our TMT quantified proteins were also found in our label-free data set. And so ultimately, this entire workflow allows us to, in a very sample-type-agnostic manner, process and quantify proteomic profiles across multiply differentially treated samples. We're able to reduce the overall amount of starting material required by over 250-fold. We decrease the amount of TMT reagent required by 12.5-fold, which leads us to a greater than 14-fold increase in mass spec throughput, so we can give our collaborators go from plate to data in just 3 days. And so to kind of summarize our entire talk, this is now our automated high-throughput proteomic sample preparation platform at NCATS, where we're able to now successfully process adherent cells, serum, and C. elegans. Our lysis can be done the traditional way using our Bravo instrument, or using now our Covaris instruments. We can then go on to do a reduction, alkylation, digestion, as well as TMT labeling on the Bravo instrument, allowing for quicker time to get our data and to move on to follow-up experiments. And what we're really excited about is bringing in this, the Covaris R230 instrument, is that it is really capable of working with much more difficult sample type. So we're excited to now further expand this platform to treating or to processing tissues, 3D organoids, or suspension cells. And with that, I'd just like to acknowledge our mass spec team at NCATS. We definitely want to shout out our collaborators that process all of our C. elegans samples, did a lot of the manual isolating of these very minute C. elegans, as well as the entire Covaris team, specifically Melissa, Paula, Tom, and Samir for all their help establishing these workflows. And we'd be happy to take any questions. And I will be giving a poster today, so happy to continue the conversation there as well. Thank you.